So, so we have already explained in the last class about the cellular foundations. We have discussed about the various components in the cell, and in case of prokaryotes as well as eukaryotes. Then we discussed about the classification of organisms. Then we discussed about the detailed structure of the bacterial cell, gram positive, gram negative, and then the eukaryotic cell and various subcellular organelles present in the eukaryotic cell. Then finally, we discussed about the supramolecular complexes that are formed by the several monomeric units in the eukaryotic cell. And then continuation with that, in this class, we will discuss about the chemical foundations. So the chemical foundations, we have already said that biochemistry is mainly aims to explain the various biological functions or the various biological form and functions in terms of chemistry. So to understand that, since the biochemistry deals about the interaction of various molecules, all these biological molecules are interact with each other. So that's why, so the understanding the chemical foundation is also important. And and we know that the in late 18th century, chemists had concluded that the composition of living matter is different from the inanimate world. So that means they said that the composition of the living matter is different from the non-living matter. Then in late 20th century, and many scientists have investigated the glucose breakdown, that is the conversion of glucose to pyruvate that takes place in the glycolysis. So scientists have investigated the glucose breakdown in yeast and also in animal muscle cells. Surprisingly, they found that they have the remarkable chemical similarities. In the sense, the glucose breakdown, whether it is in yeast or animal tissue, the same metabolic intermediates, same enzymes, and the same steps are involved. That's why, so they found that they have the remarkable chemical similarities about the glucose breakdown both in yeast as well as the animal tissues. So this indicated that, and after that the other many other scientists also investigated various other biochemical pathways, and analysis of various biochemical pathways in different organisms showed that many biochemical pathways are similar in different organisms, in the sense like for example, like we said that the glucose oxidation from glucose to pyruvate takes place in 10 steps, whether it is in yeast or in the animal tissue. In the same way, the investigation of many other biochemical pathways also revealed that all these biochemical pathways are similar in different organisms. And this similarity has been summarized by the Jacques Monod as the biochemical unity. So in the sense, these similarities of various biochemical pathways in different organisms is known as the biochemical unity. And in 1954, Jacques Monod first time said that what is true of E. coli is true of elephant. That means all organisms share a common evolutionary origin because they have the universality of the chemical intermediates as well as the transformations. You can see here that whether it is in E. coli, or whether it is an elephant, or whether it is in yeast or in other animal, the conversion of glucose to pyruvate takes place only in 10 sequential reactions, and it is same in almost all organisms. That's why most of these organisms have the same chemical intermediates and the intermediates in various biochemical pathways, and this phenomenon is termed as the biochemical unity. And later, uh, so uh, so in the cell, as we have already said that in the cell there are many elements are essential for the cell for its life, and less than 30 naturally occurring chemical elements are essential to the organism, and most of these elements have the low atomic number. And you can also see here these elements are also classified, the elements that are essential for the organisms are classified into bulk elements as well as the trace elements. And these bulk elements are mainly involved. You can see here these bulk elements are in orange, light orange color, whereas the trace elements are in yellow color, highlighted in yellow color. And the bulk elements are mainly essential for the structural component of the cell. And the most abundant bulk elements are the hydrogen, oxygen, nitrogen, and the carbon in living organisms. And that means the hydrogen, oxygen, nitrogen, and carbon, all these four elements can make up 99% of the mass of the most of the cells. So that is the importance of the bulk elements. Coming to the and coming to the trace elements. Trace elements, as we said, that these trace elements are essential for the function of specific proteins, and because mainly the hemoglobin and many enzymes require these trace elements. And these bulk elements are generally required in grams in the diet, whereas the trace elements are required only in the milligram amount in the diet, because as I already said that trace elements, mainly the iron, copper, zinc, these trace elements play an important role in for the function of the specific proteins, mainly enzymes and the hemoglobin and so on. For example, if you take the hemoglobin, we know it is a tetramer, and it has two alpha subunits and two beta subunits, and its main function is only to carry the oxygen from lungs to the tissues, and also to carry back the carbon dioxide to the from tissues to the lungs for the exhalation. And if you look at this structure of the hemoglobin, and it has the globin part as well as the heme part, and the heme part has the 4' ring system. And since it is a tetramer, each subunit has the one heme pocket, and that the heme pocket is composed of the 4' ring system, and this 4' ring has the iron in its +2 oxidation state. So that's what these trace elements, mainly iron, copper, zinc, plays an important role in the specific function of many proteins. Here, in this case of hemoglobin, the iron is the site for the oxygen binding. So without the Fe2 plus, the hemoglobin cannot carry its oxygen and the oxygen in the circulation. So that means the iron that is present in the protein plays a very important role, especially. And of course, you can also see here that the iron that is present in the hemoglobin constitute less than 0.3% of its total mass. That means it is present in a very negligible amount compared to the concentration of hemoglobin in the red blood cell. And so in the same way, these trace elements are also important for the function or the catalysis of many enzymes, and most of the enzymes use a non-protein component for their catalytic activity, and this non-protein component is known as the cofactor. And some enzymes use organic cofactors, some enzymes use inorganic cofactors, and depending upon the metal ions, if the enzyme uses the inorganic cofactor, these inorganic cofactors are mainly the metal ions, and depending upon the metal requirement, these enzymes that uses metal ions are also classified as metal enzymes, metal dependent enzymes, metal activated enzymes, and so on. And in metal enzymes, the metal is the integral part of the enzyme, similar to the iron in the 4' structure of the hemoglobin. And you can see here the Zn+2 is a cofactor for the carbonic anhydrase and alcohol dehydrogenase, and Fe+2, Fe+3 cofactor for the cytochromes, and also hemoglobin, and the pyridoxine, and copper Cu+2 and Cu+ is a cofactor for the cytochrome oxidase, and magnesium and is a cofactor for the pyruvate phosphokinase. So in all these these metal ions are integral part of the enzyme. So in the sense they play an important role in the catalysis. Without these metal, the enzymes cannot catalyze the reaction, that is the conversion of substrate into the product. So that's why all these trace elements, not only the bulk elements are mainly involved in the structural component of the cells and tissues, whereas the trace elements are essential for the function of various proteins and enzymes. And then coming to the cell also has many biological macromolecules, and these biological macromolecules are mainly composed of the carbon. And these if you see that the cell has many biological macromolecules, all these biological macromolecules are mainly composed of carbon and with different functional groups. And this carbon that is present in the cell can, or in the biological molecules, can form either a single bond or the double bond or the triple bond with hydrogen and the oxygen and the nitrogen and so on. So carbon can form single bond with hydrogen, double bond with oxygen, and also triple bond with nitrogen and so on. That means the carbon can form different bonds with this one. And in addition to this, the carbon can also form 4 stable single bonds with other carbon atoms, and so that means it has this formation of carbon with 4 single bonds with another carbon has the greatest significance in the biology. And then if the carbon is attached to the 4 different atoms, and that gives the tetrahedron arrangement, and this in this tetrahedron arrangement, the angle between any 2 bonds is 109.5 degrees, and the bond length is the average bond length is 0.154 nanometers. And since the single bond is in the carbon single bond, in this tetrahedron rearrangement, since they are the single bonds allow the rotation around the axis. Whereas if the carbon double bonds are the present, and the carbon double bond has a shorter bond length, and also they are the rigid, and the bond length is 0.134 nanometers. So you can also see here that if it is carbon single bond, its length is 0.154, and if it is carbon double bond, it's the bond length is shorter, that is 0.134. And the double bond generally doesn't allow the rotation, in the sense it allows only the little rotation, because it restricts the rotation of the atoms around the its axis. And so then this carbon can also form the linear chains, branched chains, and cyclic structures in the biological molecules. So whatever we see, the proteins, carbohydrates, and other lipids, the carbon that is covalently linked in the biological molecules can form linear chains and branched chains and the cyclic structures. And that's why in the evolution, carbon compounds were chosen for making the molecular machinery of the organism. And no other element other than carbon can form such a different size, shape, and the molecules of different size, shape, and the composition. That's why the carbon compounds were selected for making molecular machinery during evolution. So no other elements can form such a diverse compounds. And now, so coming to the most of the biomolecules that are present in the cell, most of the biomolecules are the derivatives of the hydrocarbons. In all these hydrocarbons, the hydrogen atom is replaced by a variety of functional groups. So as a result, we get the various organic compounds such as alcohols, amines, aldehydes, ketones, and carboxylic acids. And you can see here that when the hydrogen atom is replaced with any different variety of functional group, we get different class of molecules, and all these are the some common functional groups present in the biological molecules like methyl, ethyl, phenyl, and of course so on. All you, I think you have already studied carboxyl and the ester, ether, aldehyde, amino, amido, imine, and all these are the different functional groups that are commonly present in the biological biological molecules. And these functional groups are very important for interacting with the in the various interactions in the cell. And many biological molecules are polyfunctional, in the sense they have more than one functional groups in their structure. For example, if you can see here the coenzyme A, this coenzyme A is a carrier of acetyl groups in various enzymatic reactions, and this is the structure of the coenzyme A. You can see here it has many functional groups. So thioester, amido, and hydroxyl, methyl, phosphoanhydride, imidazole, and amino, and phosphoryl, and so on. So that means in all hydrocarbons, the hydrogen, when the hydrogen is replaced with different functional groups, various organic compounds are formed like alcohols, amines, aldehydes, ketones, and carboxylic acids. But many of the biological molecules have more than two or more types of functional groups, and these functional groups are very important for its reactivity and its interaction in the cell. And then coming to the various molecules that are present in the cell. The cell contains various small molecules as well as the macromolecules. And the small molecules, they are mainly present in the cytosol. From 100 to 200 different small organic molecules are present in the cell with a molecular weight of 100 to 500 daltons. They are all very small molecules. These small molecules are mainly amino acids, nucleotides, sugars, and the phosphoryl derivatives, and the mono, di, and the tricarboxylic acids. And these molecules are either polar or charged, and they are water soluble, and they are present in the cell in the micromolar to the millimolar concentration. And since all these are amino acids, nucleotides, sugars, all these are phosphorylated derivatives, all these are polar molecules. So therefore, they are trapped inside the cell. They are trapped in the cell because they cannot cross the bilayer. As I already explained in the yesterday's class, that the lipid, the plasma membrane is the lipid bilayer. Lipid bilayer, only hydrophobic molecules can cross, whereas the gaseous molecule can cross, whereas very small, the polar uncharged molecules can cross, but the polar molecules and the charged species cannot cross the bilayer. In order to transport these molecules, of course, the cell is equipped with various mechanisms by having the channels as well as the transport proteins. You can also see here, this is the lipid bilayer, and the all these molecules, the polar molecules, are trapped in the inside the cell, and where they cannot come out. And in order to transport such molecules either in and out across the membrane, many specialized transport mechanisms are available. These are the ion channels as well as the active and the passive transport mechanism. And these are and in addition to the amino acids, nucleotides, sugars, and the phosphoryl derivatives, and some certain cells also have the specialized molecules. For example, the plant cell, vascular plants contain specific specific molecules. Those are known as the secondary metabolites, and these secondary metabolites mainly in morphine, quinine, nicotine, caffeine. All these are secondary metabolites present in the plant cell, and these secondary metabolites, plant uses these secondary metabolites for a specific function, but these secondary metabolites are also various uses for therapeutic uses for humans and humans. So that means the cell has 100 to 200 different small organic molecules, and these are mainly amino acids, nucleotides, sugars, and the phosphoryl derivatives. They are polar, are charged, and they are water soluble. Hence, they are trapped in the cell because of the hydrophobic nature of the bilayer, and in order to allow the movement, in order to facilitate the movement of these molecules across the membrane, the cell is equipped with plasma membrane is equipped with various mechanisms by having various transporters. And in addition to these primary metabolites, the cell, some organisms also have the secondary metabolites. These secondary metabolites, especially in the plants, the morphine, quinine, nicotine, and caffeine are the secondary metabolites. In addition to this, the certain bacteria also produce antibiotics, and they are also the secondary metabolites. The secondary metabolites, these organisms produce the secondary metabolites for a specific function, but the humans use the secondary metabolites for various therapeutic applications. And this is about the various small molecules present in cell, and the total the total number of the small set of molecules present in a cell is known as the cell's metabolome. The total small molecules present in the cell is called as the cell's metabolome. Metabolome means total metabolites of the small molecules present in a given cell. So this is about the set of small molecules present in the cell. So coming to the macromolecules. So the most of the biological molecules present in the living cell are the macromolecules, and these macromolecules have the higher molecular weight. Their molecular weight is greater than 5,000 daltons, and these macromolecules are assembled from the simple monomeric units or the simple precursors, and if the short polymers are called oligomers, and the synthesis of the macromolecules from their simple precursors is always a energy consuming process, because it is a anabolism is always energy consuming activity in the cell, and these macromolecules are also further assembled into the various supramolecular complexes. These supramolecular complexes, as I already said in yesterday's class, the eukaryotic cell has supramolecular assemblies, and these supramolecular assemblies further in turn forms various functional units such as ribosomes, and means the many biological molecules that are present in the cell are macromolecules. They have the higher molecular weight. They are assembled from the simple precursor molecules, and since the formation of the macromolecules from the simple precursor molecules is always a energy consuming process, because it is a anabolism, and these macromolecules are further assembled into the supramolecular complexes, which in turn forms various functional units in the cell, such as ribosomes and so on. So, and you can also see here that the major components are the major the molecules present in biological molecules present in the cell. You can see here that E. coli cell has the 3,000 different proteins, and also it has many other different types of polysaccharides, different types of lipids, and various monomeric various intermediates, and as well as the inorganic ions present. So this is the for the in case of E. coli, and the coming to the the biological macromolecules, the major important biological macromolecules that are present in the cell are the proteins and the nucleic acids and the polysaccharides as well as the lipids. These are the important biological macromolecules present in the cell. And we will see the one by one in detail. And we know proteins. First, we know the proteins. These are the polymers, and they are made up of with several amino acids. When the amino acids are connected to each other by the peptide bond, proteins are formed, and these proteins have various functions in the cell. I think many of you know, and they have the transport function. They have the the you can see here they have the structural function, storage function, and so on. And you can see here that, for example, the hemoglobin is a protein, and hemoglobin has the transport function. It carries the oxygen. And whereas if you see the myoglobin is a storage protein that stores the oxygen. So that means myoglobin can also be example for the storage function. In the sense, then some proteins have the contractile function. For example, actin and myosin mainly involved in the muscle contraction, and the some proteins have the structural role, mainly the collagen and the keratin. All these are the involved in the present in the tendon and the cartilage and so on. The keratin is present in the hair, skin, wool, and the nail. And some proteins are also the hormones. For example, the insulin. Of course, so it is not a. It is a insulin has it is a it has two chains, A chain and B chain, and this insulin is also involved in the regulation of the glucose concentration in the cell. And many proteins are enzymes except ribozyme, RNA acting as a catalyst. They catalyze various biochemical reactions, and the proteins are also have the protective role. For example, immunoglobulins. Immunoglobulins are proteins. They they are involved in defense mechanism. So that means proteins have various roles in the cell. So starting from the structural, contractile, transport, storage, and catalysis, as well as the defense mechanism. So that's why the proteins are important biological macromolecules in the cell. And the the sum of the all proteins present are functioning in a cell is known as the cell's proteome. Cell's proteome. If you take any cell, the total proteins functioning in a cell is known as the proteome. And the proteomics is the study of characterizing, or the study of large scale of proteins is known as the proteomics or the proteomics. It is a technique that is to study the large scale of proteins. So this is about the proteins, and in summary, these proteins are the polymer of amino acids. These amino acids are connected to one another by the peptide bond, and details we will be discussing in the third, fourth module under the proteins. And the second biological macromolecules is the nucleic acids, and the DNA, RNA is the nucleic acids. They are the made up of with the nucleotides, and you can they are the polymer of nucleotides. We know the nucleotide has the nitrogenous base, sugar, as well as the phosphate group. You can see here, and all these nucleotides are connected by the phosphodiester bond. And this DNA and RNA, they are the nucleic acids. They have they store the genetic information and transmit the genetic information. And some RNA molecules, like as I said, the ribozyme also has the structural as well as the ribozyme has the catalytic role. Some RNA molecules have only the structural role. And the come here the genomics here, the total DNA, or the entire sequence of the cell's DNA is known as the genome. And the genomics is the study of the whole genome of the organism. That is the characterization of structure, function, evolution, and mapping of the genomes is known as the genomics. So then coming to the third class of the biological macromolecules is polysaccharides. These polysaccharides are the polymers of simple sugars. You know here, and when the monomeric sugar units are connected to each other by the glycosidic bond, and polymeric polysaccharides are formed, and cellulose, starch, glycogen, chitin, all these are polysaccharides, and some polysaccharides have structural role, and some polysaccharides have the storage function, and so on. We know that the cellulose is the structural component in the plant cell wall, whereas the starch is the storage polysaccharide in the plants, whereas the glycogen is the storage polysaccharide in the animals, whereas the chitin is mainly involved in the structure, mainly present in the structural components of various crustaceans and so on. And so that means these polysaccharides have various roles like storage function and as well as the structural role. And in addition to this, these the saccharides that are present on the cell surface, you can also see here saccharides that are present on the cell surface, they are also known as the glycans. Glycans means they are the carbohydrate polymers. The saccharides that are polysaccharides that are present on the cell surface, in the sense they are medium chain length carbohydrate polymers, they are known as the glycans. These glycans, the polysaccharide, these glycan chains that are present on the cell surface has various functions such as cell-cell interaction, cell-cell interaction, cell-cell recognition, cell-cell adhesion, and host immune, host pathogenic interaction, and then the blood clotting and the blood group formation of various blood group substances. In all these cases, these glycans play an important role. You can also see here that you know very well about the human influenza virus. This is a structure of the human influenza virus, and this virus has many epitopes on the surface, and but the two epitopes are very important for the human influenza virus to to continue its life cycle in the host cell. And these two epitopes are the neuraminidase and hemagglutinin. And here the hemagglutinin, recognizes the host cell through a sialic acid residue. You can see here, this is the glycan chain, in the sense a carbohydrate chain that is present on the cell surface. And so this hemagglutinin, that is epitope that is present on the influenza virus, recognizes the sialic acid residue, and further this neuraminidase breaks this linkage, and this virus then enters into the host cell. That means here, as I already said, that the sugar, the chains, the carbohydrate chains or the glycan chains that are present on the cell surface has various functions. They're not, because these glycans, in addition to be the storage and the structural components in various organisms, and these glycan chains have various roles, that is the cell-cell interaction, cell-cell recognition, and and so on. And so the best example is that the influenza virus, the recognizes the host cell because of the presence of the sialic acid, which is a glycan that is present on the surface of the host cell. And the sialic acid is a modified sugar actually. Yeah. So in the same way, there are even human influenza virus also uses recognizes the host cell using various the heparan sulfate proteoglycans present on the host cell. And of course, we are not here to explain this. And I just wanted to convey the message that polysaccharides, they are not only the sweet molecules, and they have various roles, not only the storage of fuels and the structural components. Beyond that, the polysaccharides or the glycan chains present on the cell surface has various functions. So that is the interaction of the cell, one cell with another cell, or recognition of one cell by the another cell, and cell-cell adhesion, and so on. And so this you can also see here that the cell-cell interaction or the cell-cell recognition and the interaction between the hormone and the cell are all these or interactions are mediated by the carbohydrate, the glycan chains present on the surface of the cell. So that means the glycans have various roles in the cell. So this is about the polysaccharides and third class of macromolecules. Then the fourth class of macromolecules is the lipids. And we know the lipids are the hydrocarbon derivatives and they are insoluble in water, and these lipids are mainly these lipids are mainly present in the membranes. In the sense, biological membranes are lipid bilayers. So that means these lipids are the components in the membranes. And in addition to this, the excess of energy is also stored in the form of triacylglycerol in the human body. And these are also triacylglycerols are also the storage energy rich storage fuels. And some lipids are the pigments, mainly the carotenoids, and some lipids are involved in the signal transduction pathways, and they are also involved in the hormone synthesis and bile acid synthesis, and the lipids, mainly the cholesterol, that is a derived lipid, is the precursor for many male and female reproductive hormones, mainly aldosterone, cortisol, estradiol, progesterone, testosterone, and pregnenolone, and see, all these are derivatives of the cholesterol. They are derived from the cholesterol. So that means lipids also not only the components in the membranes, and they are the energy rich storage fuels, and they also have the function in the signal transduction, and they are the precursors for the mainly the dietary lipid. Cholesterol is the precursor for the many reproductive hormones, the reproductive hormones in male and females. And the the total lipids present lipid containing molecules present in a cell is known as the lipidome. So we have already introduced the three terms, and the one is the metabolome. Total the set of molecules present in a cell is known as the metabolome. Then we said that the proteome, that is the total proteins present in a cell, or the all the proteins functioning in a cell, is known as the proteome. Then the entire sequence of DNA, cell's DNA, is known as the genome, and the total lipid containing molecules present in a cell is known as the lipidome. And then all these biological macromolecules, they interact with each other in the cell, and the interaction is mainly the function of many biological macromolecules, mainly proteins, and proteins are mainly dictated by its three-dimensional structure. And this three-dimensional structure is described by the conformation and the configuration. And we know that the many carbon containing compounds generally exist as stereoisomers, in the sense they have the stereoisomers. We know that they have the same chemical bonds and the same chemical formula, but the different orientation or the arrangement of atoms. And these stereoisomers can be represented with either a structural formula or the ball and stick model or the space filling model. And these stereoisomers, so in the human body, and only particular stereoisomers are active, and these stereoisomers are also again classified into the enantiomers and the diastereomers. Enantiomers are the stereoisomers that are mirror images of each other, and that means they have the same chemical formula, but they different orientation. They are the mirror images of each other. They are known as enantiomers. Diastereoisomers are stereoisomers which are not mirror images of each other. And all these biological molecules that are present in the cell are stereospecific, in the sense they interact only with the interact specifically. So in the sense interaction is highly specific, only, for example, if you can see here that and these stereoisomers also have the different functions in the different effects in humans. For example, you can see here, this is the aspartame. Aspartame, L-phenylalanine methyl ester has the sweet taste, whereas the D-phenylalanine methyl ester has the bitter taste. So look at here, the aspartame, the structure is same. In one case, this is a L-phenylalanine is present. In another case, D-phenylalanine is present. So that means these properties are also dictated by the stereo specificity. That means stereoisomers have different properties as well. In the same way, and in the same way, if you see the amino acids that are present in the proteins, so we know that proteins are the proteins are built with the 20 standard L amino acids. Only L isomers are present in the mainly proteins. In the same way, the D sugars are mainly present in the cell because they are the naturally abundant, they are the predominant. That means the cells also use only particular isomer, only one type of isomer, and so coming, this is about the chemical foundation of the biochemistry. In summary, the carbon, as I already said, that in the cell, the many chemical compounds are present, and all these chemical compounds are the derivatives of the carbon, and this carbon can form different types of molecules and the carbon has the ability to form single bond, double bond, and triple bond, and the four bonds either with the other carbon or with the oxygen or the hydrogen or with the nitrogen. And these carbon, because of this nature, the carbon compounds were selected during the evolution for the formation of different structures of different molecules of different size and shape. And the cell has, if you see that the cell has both the small set of molecules as well as the macromolecules, and the small set of molecules are mainly classified into the bulk elements as well as the trace elements, and the bulk elements are mainly involved in the structural function, whereas the trace elements are mainly involved in the enzymatic function. And the coming to the most of the biological molecules are macromolecules, in the sense they have the higher molecular weight, and these macromolecules are mainly the proteins, nucleic acids, and the polysaccharides and the lipids, and all these molecules functions in the cell. And the property of the macromolecules are mainly dictated by its three-dimensional structure and the three-dimensional shape, and only because many biological molecules are active only in one isomeric form, because as we have already seen that only in the cell only L amino acids are present in the proteins, whereas in the cell the D sugars are mainly abundant in the cell, because the cell also uses only one type of isomer. So, and the interaction of all these biological macromolecules are highly stereospecific. As I already said, that all enzymes of the carbohydrate metabolism acts only on the D sugar. All enzymes that are present in the amino acid, all enzymes that are involved in the amino acid metabolism can only act on the L amino acids, not the mainly the D amino acids. That means they are the interaction of the biological macromolecules are highly stereospecific, in the sense they can act only on a particular isomer. So this is about the chemical foundation. In next class we will discuss about the physical foundations.